Hello and welcome to another episode of Momento Malum today. The sugar beet is a crop of primary economic importance in Europe. This species accounts for 25% of the world's sugar production. The basic requirements for any crop improvement program is genetic variation for traits of interest. Therefore, a proper assessment of the gene pool and structure of the beet population is needed for the organization of breeding material as well as the use of genetic resources for crop improvement. Dr. Lee would use SSR marker technology to further understand the beet gene pool. This technology had been previously widely used for population genetic analysis in beets. Most of these studies have been used to examine the natural crossbreeding between cultivated and wild beet populations, the genetic diversity of wild beets, and the relationship between beets and other species. There is, however, not been any study prior to Dr. Lee's work on the gene pool of the elite sugar beet germplasm, and that gene pool's genetic diversity. To analyze the gene pool, a total of 289 sugar beet inbred lines were analyzed, and here are the results. The results of the study found that the gene pool of the analyzed lines was split into 3 groups, 2 of which had 3 subgroups. Group 1 consists of 115 genetic lines, Group 2 consists of 106 genetic lines, and Group 3 consists of hybrids between the above-mentioned groups. Group 1 is further broken down into 3 subgroups. 2 of these are genetically distinct subpopulations that are of pure Group 1 pedigree, with a third group containing minor traces of Group 2 pedigree. And the same thing applies to Group 2. Among the subgroups, it's found that Group 1 subgroup A and the populations in Group 2 with minor traces of Group 1 pedigree had the greatest level of genetic variation within these groups. The discovery of 2 distinct genetic groups was surprising to the researchers, because the entire collection is derived from a single population created in the 70s. It is speculated that the years of recurrent reciprocal selection, in which populations are selected to genetically complement each other, while at the same time fixing genetic traits and preventing intermixing between populations. It is believed that this process led to the creation of differentiated populations. It was also found that the overall level of genetic diversity in its population is 60%, which is surprisingly similar to the feral beet lines. The Group 1 lines were found to be more genetically diverse than the Group 2 lines. It is speculated that this is because these lines are older and derived from the original beet population created in the 70s, while the Group 2 cultivars are derived from a subsample of the Group 1 lines, making that group less genetically variable. Dr. Lee concludes by stating that the Group 1 cultivar should be prioritized in the creation of new inbred lines in F1 hybrids. And that about covers everything. Thank you for watching. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below.